So welcome to this open house of biointegrated design, the MArch and the MSc programs. I'm Marcus Cruz. I'm Professor of Innovative Environments at the Bartlett School of Architecture.
I'm Brenda Parker. I'm an Associate Professor of Sustainable Bioprocess Design at the Department of Biochemical Engineering.
And Nina, maybe you want to introduce yourself.
Hello, everyone. I'm Nina Jotanovic. I'm a lecturer in climate change and biodiversity for biointegrated design.
So we want to share with you a few thoughts and ideas and explain a bit the structure of the BioID programs. As you can see on this image, we are very interested in zooming in and understanding the complexities that we are surrounded by and the biochemical processes that explain how things grow, how they propagate, and how life on a very small scale, but also on a very large scale, really has a profound influence on who we are. Of course, we are having a great influence on what is happening also with these species. So there's a total exchange. Now, what is fascinating about these species that we are looking at is that they're poikilohydric. These are mosses, but there's a lot of algae and cyanobacteria, and these are microscopic images of how these consortia of species coexist. And there is a lot of fascinating, interesting complexity in what is happening there. The poikilohydry that I mentioned is this capacity of certain species to switch on and off the photosynthesis. And that is that depending on the availability of moisture, they can respond. So this bioresponsiveness is something of great interest for our program, and how we can design and integrate these phenomena in a scale-up process from the petri dish to the scale of the built environment.
So from the micro scale to the macro scale, another place where there's fascinating interactions happening is here at University College London. So you may be aware that we are a university that's very highly ranked for architecture, in fact, #1 in the UK and also in the world. And in terms of biochemical engineering, we are the first university in the world to teach the subject. And indeed, we are the first university in the world to bring together the departments of biochemical engineering and architecture to produce such a program. And what this means is that we've got a really interesting interdisciplinary landscape here at University College. And this enables us to run a program that is highly experimental, such as BioID. We're very fortunate. We're not actually based at Bloomsbury. We're actually based out here at Here East, which is on the Olympic Park. So this is the former comms building of the 2012 London Olympics. And we have transformed this building to host not only our workshops, but a laboratory and a studio environment. And it's home to several other interdisciplinary master's programs as well. So it provides a really rich interactive environment with state-of-the-art facilities for designing and making. And here you can see us with Oren Katz, who you may recognize, in the BioID lab. And it was really important to us to create a dedicated facility, a lab facility, which is actually for people who are designers. And the whole idea is to create this interface between the lab, the studio, and the workshop environment, where we're creating prototypes, we're testing them out, we're inoculating them with organisms, and we're engaging in this sort of design, build, and test cycle where we're able to rigorously apply laboratory protocols, but also engage with the tools of scaling up and manufacturing.
We do spend a lot of time in the studios where project reviews and discussions are happening, apart obviously from tutorials and other sort of peer-to-peer discussions. It's very important, the physical aspect of what students do in BioID. We do really focus a lot of times on the materials, the properties, the performance of materials and species, but also how on a more structural level, on a tectonic level, these materials can be fabricated. And we use advanced tools for that. So it's always very special for us when everybody puts pretty much everything on the tables, and we can sort of discuss really how this work can lead ultimately to some quite novel solutions.
And well, in order to look into these different scales, as was mentioned at the beginning, and to tackle the issues from various perspectives and disciplines, also our core team is, there's a certain ecology of people in the team that is really necessary for you and for the future students to become interdisciplinary. So here are some of the core staff at BioID, and as you can see here, some of them are really having a background in architecture, while other in biology, biochemical engineering, and some of them are more focused on environmental design or on some other digital techniques and fabrication techniques, as Marcos was mentioning, that physical aspect of the work is a very important one.
We maybe should very briefly introduce the structure of the 2 years. These are 2 programs, the MArch and the MSc. They run in parallel, they run in tandem. Students work together and form teams, collaborative teams conjointly. And this is, as you know, a 21-month long system, whereby we have a first year, which is structured in the first term with 4 modules. The 4 modules are mainly focused on the first one on an introduction to design. It's a sort of the preliminary design tests and experiments. Then we have a literature review, which gives you basically an understanding of how to structure research, how to read basic literature. And there's a paper that students have to write, a short one. Then there are the computational skills. This is a key module also to learn how to use advanced software. And there's a variety of different work packages that students learn how to work with. And then there is the laboratory and science methods module, which introduces students, especially those who come from a non-science background, in learning how to apply science protocols and understand the laboratory, how it works. This module also allows students to be independent in the laboratory, which is very, very important for us. In term 2 and 3 into the summer, we have 2 modules in the first year. The main module, which is 75 credits, is the main project module, which is basically continuation of design. There's a synthesis of many of these previous learnings that students integrate. It's collaboratively developed, this project, in all these months. And it's really the first big research project that the students engage with. And simultaneously, they have this thesis report, which is a smaller module, but very pivotal, where students get tailored tuition by experts from within the school or UCL, or even outside UCL, to develop a very particular individual piece of work that feeds into the collaborative project. Now, in this second year, the first term is pretty much an introduction to the main project. And there's also running in parallel to a specialization that students go through. So students have done a lot of interdisciplinary work in the first year. They have tried out many things. And then the first term of year 2, they want to specialize, they want to focus. They start developing the main project of the year with their teams. And then in the second and third term until July, they develop and give a final resolution to the main project that they develop, which is also the biggest module of the entire course. And as you can see, it's 180 credits in the first year, 120 credits in the second year, altogether 300 credits. It's a very intense educational program. So there's many contact hours between tutors, this ecology of people who we have around, and seminars, workshops, etc., and other activities that students have in order to get sufficient input.
Yeah, maybe it's worth saying this is also a full-time program. So you have access to Here East, actually 7 days a week if you wish, but we're teaching Mondays to Fridays. As Marcus mentioned, we have a kind of very varied program of workshops, practical-based teaching, but also you're invited to engage in quite a lot of self-driven learning. So you'll notice here that although we have a lab module, it's about introducing you to the methods and scientific practice. And we don't teach a biology 101 module. You're not expected to sit an exam and know off by heart kind of DNA and cellular architecture and structure. We're much more interested in teaching you how to apply the tools of biology. And the best way to do that is through practical work. It's through designing your own experiments, and it's by learning how to grow things. So for those of you who are maybe coming from a fine arts or an architecture or a design background, this is a chance for you to really get into the lab and start to apply the tools of biology. And we're really trying to make this as welcoming as possible, but it is, of course, a steep learning curve. And for the MSc students, we're not attempting to recreate an architecture program. We are teaching through a series of kind of targeted sort of sessions and seminars how to apply the tools of design as well. So this is really fundamental and core to the spirit of how we teach on the program, that it's also very much a journey of discovery and creating your own knowledge, which is, you know, in a kind of program like this, a wonderful opportunity to make use of all of the resources that UCL has to offer as well. So perhaps just to give you a flavour of some of the things that have been happening, we took a bit of a diary snapshot to say, you know, what have we been up to recently? So we've had, obviously, because we're in London, we're really fortunate to have just some terrific people passing through the city. So people like Duncan Cameron teaching us about sort of soil biology and soilless agriculture. Barbara Imhoff talking about space and algal cultivation and futuristic architectural environments. We've been on botanical walks with Mark Spencer, who's a forensic botanist, learning much more about the ecology that surrounds us. We complement that with kind of very intense, perhaps technical coaching sessions where you might be learning a specific skill, for instance, like film and Unreal Engine, to be able to realize and visualize work. We also engage in quite a lot of discussion around the philosophical and critical thinking that's required. So we have a forum that we call the Salon, where we engage with perhaps contemporary topics that are coming up in this interdisciplinary field, but also examine our own practice and look at how we can make it equitable and diverse, and also equip you, perhaps the future teachers and leaders as well, to be sensitive and receptive to some of the issues that are emerging. Students have plenty of opportunities for exhibition and engagement with the public. So we have had an open house this last 2 years as part of the London Festival of Architecture. And students also exhibit at the BPro show at the end of the first and the second year. So this hopefully gives you a little bit of a flavour of just the variety and the profile of the kinds of people who come to speak and teach our students. And really, every year it varies. And this means that every year the outputs of BioID are interesting and enriched by a different set of people who are passing through.
The students who have a design background, so who did not do science before, they have a cultivation workshop. They also obviously have the module where they learn how to work in a lab and how to apply science methodologies. The students who come from the sciences, who have never done design before, they obviously have the introduction to design, in the first term, but I also give them a series of seminars to introduce them to design practice. So design practice, and also to key references, the cultural portfolios, etc. So there is in the first term very much, and throughout the first year, a sense that students who are coming from such diverse backgrounds, they require additional support in order for them to become much more equivalent in terms of how they operate later on in the second year. Oops, sorry.
So we wanted to maybe situate some of the work that happens within BioID in terms of what really is one of the most pressing challenges that we face. And that is our growing climate emergency. And we often talk about this in very high-level terms in terms of carbon. But there was a very interesting paper published just this year about the 9 planetary boundaries and where we are operating outside of a safe space in terms of preventing or being able to sustain future generations. We know that we're emitting chemicals that are accumulating in our atmosphere and in our soils that we're unable to kind of break down or degrade. We know that, of course, we are facing ever-growing levels of carbon dioxide, but we also need to look at nutrient flows, so nitrogen and phosphorus, and where we are also not recycling nutrients in an appropriate manner, and how we safeguard things like fresh water so that we will have enough for everybody going forward. Oh. Marcus is preventing us from getting radiative force, and so that's good. But needless to say, this is the level of nuance that we want to start engaging with the climate emergency and BioID. It's not to fall into this doom loop that it's all too difficult and we don't know where to start. It's to actually break it down into things that are very meaningful. And we're really fortunate. We have an incredibly diverse group and cohort within BioID, many nationalities, people coming from all over the world to also share and interact and share their experiences as well, which enriches the discussion and also helps us tackle this problem with a great deal of nuance and understanding and cross-cultural understanding of what's happening.
So we talk a lot about the Anthropocene legacy of what we humans are doing. And as Brenda was mentioning, on the one hand, one can see it from a very sort of negative perspective, and that's important, is to learn from the mistakes that we have made, from the negative impact that currently our habits are having on the planet. On the other hand, there are opportunities as well that emerge. And what you can see here, when you look at the skyline of London in construction, is that we humans have added a phenomenal amount of surface area to the world. And these surfaces, if they're really designed and thought to take part in the cycles that we have in the environmental cycles we have on our planet, then we could really make a difference. So in a way, we have built with materials that are very unsustainable in the built environment. Yet if you start thinking that the future might look very different, we will work and build with biomaterials that have totally different qualities, and some of them are starting to emerge already, then this whole surface area, this grand jungle that we have produced, is starting to offer phenomenal chances for us to do things very differently. And we are very curious about things like what you're currently looking at on the slide, which is when a student of ours from Saudi Arabia started looking very carefully at the desert crusts. And you would think that there's pretty much no life in it because it's so hot and so dry. And it's just a bit of eroded sand with some other particles in it. In fact, it's not at all like that. What she realized is that it's fully inhabitable and inhabited. And there is cyanobacteria that travels in the soil, which is quite extraordinary. So we are interested in a way, demystifying some of the things that we thought we understood, which we are now understanding very differently, and starting to reconsider what the surfaces in our world are, especially those that we humans are creating. Do you want to say something about this?
Absolutely. So this is the work of Ian Robinson, who's one of our graduates and now actually a doctoral student. And he was interested in plant callus tissue. So if you've ever grown a geranium, you might be familiar with callus tissue. This is effectively plant stem cell tissue culture. And depending on the hormones that it receives, it differentiates into roots and leaves. And we are interested in bioID in not only things that are very near term and perhaps commercial, but also the speculation and wonder that comes with engaging with novel forms of biology. Move on. And here you can see just some of the kind of fabrication work that Ian and the group have been exploring to look at how callus tissue can be, how we can deliver nutrients to the callus tissue, and the kind of forms we might create in vascular structures. And again, this is very much emerging research, which is why Ian was awarded a doctoral scholarship to continue after the program. But it also gives you a flavor of the kind of ambition, but also exploration that we want to engage in on bioID. Another former MSc student is Prantar Tumuli, and during his 2 years, he became absolutely fascinated by a filamentous cyanobacteria or prokaryotic alga called Oscillatoria animalis. And much like the story of the desert crusts, he was interested in how filamentous organisms can bind particles together. And so he continued his work and became more and more interested in the impact of light. And this also gives you a bit of a flavor of just some of the scientific research. Prantar trained originally as an architect and now has fully embraced the sciences. And what he has really discovered is the way that cyanobacteria grow towards different wavelengths of light actually impacts the type of biomaterial or engineered living material that you can produce. And this is just some of the data that he was able to generate from his research on the program. And what's, I think, really gratifying is not just to collect data and produce graphs, it's actually to make things that are informed by the knowledge that you generate on the program. And here you can see Prantar doing some robotic milling of the aggregate material that he had developed. And Prantar has been tremendously fortunate that he has also been able to progress his work again through a doctorate, but also through a patent application, because what he's doing is so novel and offers great potential for a kind of set of materials for the built environment that have unique properties, but also potentially interesting carbon footprints as well.
So a student who worked together with Prantar, I wonder whether this is a film. It should be a film. No. Oh, that's a shame.
Okay.
Anyway, this was a little film. Was Daniel Wen, who was looking much more at sort of the visionary aspect of how to apply such materials in this sort of multi-material context, and looking at translucency qualities. And in fact, we are very interested in using advanced computation to simulate scenarios that we have never envisioned before. We really need to be quite daring in how to reinvent and rethink how our future is going to not only perform, but look like and feel.
Oh, there was a film.
Maybe there was. Maybe we needed to click on it.
Oh, but nothing happened.
No, that is strange. Okay.
Okay.
So as you can see, our interest is to combine not only these different techniques and skill sets, but very much the different scales. And this is where the great challenges are lying. How do you go from something that is tiny, small, and control that, and then apply it on a much larger scale? This student, Mac van Dam, worked with cork and extracted suberin, which is the resin of cork. And he used quite interesting techniques of milling and then robotic extrusions to create these designed receptacles where you can have the growth of roots and plants in it. And ultimately, what he was trying to do was to create a new, novel, impermeable green wall. The majority of the green walls that you see around you are pretty unsustainable. First, because they need a lot of water irrigation, because they cover very large areas. And also they need a lot of maintenance. Here, the idea is to work with plants that hardly need any maintenance, but also, instead of covering an entire surface, to let light through so that you can actually have windows behind. And Mac is now currently working on creating a startup. His work also was selected for the Lisbon Triennale, where he exhibited. That's where you see him here on the left-hand side corner. And it is getting a lot of attention internationally due to some of the novel solutions in this project. We do exhibit a lot, as Brenda mentioned before. These are just some of the cases. On the top right-hand side, we see the entire team when we were at the Centre Pompidou, where we exhibited these bioreceptive concrete panels quite a few years ago. And then below you see, for instance, an image, which was actually the first exhibition at UCL after the pandemic was just getting over. And the importance of exhibitions, not only to do with how to create the layout, how to make sure that you show the right pieces, it's actually the communication and dialogue this creates with others and how you disseminate your work. We do attract lots of curious, interesting people to BioID. And you see there in the middle top image, Lord Norman Foster, who was very interested. Both Brenda and I spoke at the Norman Foster Foundation in Madrid, and Norman wanted to come to visit the lab and see more specifically what we were doing in terms of the new materials. And he's there investigating the bioreceptive multi-material concrete composites. And on the bottom left-hand side, you see Peter Cook, who gave a series of lectures to BioID. This was a few years ago. Or on the right-hand side, you see Brenda explaining to Keir Starmer, the Labour politician here in the UK, who might become the next prime minister, who knows, and also the provost of the university, and explaining to them what we are doing. And that is interesting in its own right, this curiosity and interest in the cutting edge that other people are having in the work that students and researchers in BioID are producing.
So each year we offer a field trip to our students. Here you can see us, you can guess, we're in the north, we're in Glasgow, and in St Andrews at the Botanic Gardens. So it's an opportunity to spend a few days away for the cohort to kind of get to know each other, because obviously you have year 1 and year 2, so there's lots of peer-to-peer interaction, but to visit architectural sites and also sites of biological relevance. So we have a kind of long-standing collaboration with the Botanic Garden at St Andrews, and it's just really great to be able to take students out to these sites and be able to draw, to talk, to engage with the world around us, and to think through some of the things that are important to us as BioID.
We do take students to visit buildings. It's something that is very important to us, but it's not exclusively buildings, it's objects, it's furniture, it's stuff that is designed, that is made. You can see the Zaha Hadid building in Glasgow, also the Enrique Miralles and Benedetta Tagliabue Scottish Parliament in Edinburgh. I mean, extraordinary pieces of architecture, really interesting to visit and understand. We then went last year, actually just in this year, but last academic year, feels like it was like at least a year or 2 ago, but it was just a few months ago. We went to Florence, Siena, and Larderello. In fact, apart from the interest of Florence and Siena, and Florence in particular was very interesting to us as the Renaissance city, and how that city offered so much novelty in a certain period to the world, by contrast with Siena, which is a beautiful place that was really very much a sort of medieval place. And then, actually what we did in Florence was to get out of Florence and went onto the motorway, and there is a fabulous building, probably one of the best buildings ever built in the 20th century in Europe, which is the Micolucci Church. And you see the image there on the bottom left-hand side. It's extraordinary to visit these places and experience them together, because it gives us a conjoint experience that we then discuss and use as reference in the program. And then Larderello was, again, very fascinating, because it's a place that has geothermal qualities. It's used for energy gaining for a long time. And you walk around the landscape and you have all these fumes coming out of the ground. We were toured around by some local scientists. And that in itself was a great experience to understand how some of these geothermal properties produce certain species and define the ecologies that are growing on them. Interestingly, one of the only trees that grows around that area is the cork tree, which is the only one that can actually resist these high temperatures in the ground, not only on the surface.
Over time, I suppose, the novelty of the work and the richness of the work of BioID has been rewarded. We did mention Prantar previously. Shanil, who is one of our tutors, has also been the recipient of a number of awards. And one thing that I suppose has been really gratifying and really underlined the novelty of what we're doing here is the publication of a number of peer-reviewed manuscripts on interdisciplinary aspects around BioID, whether it's the development of a new material or whether it's the integration of fabrication and material development. All of these are hopefully ways of generating or documenting knowledge that can be also passed on to the wider community. So it's really not just about the kind of individual fame of having a paper. It's actually about making a contribution to knowledge and remembering really intrinsically what we're all here for, which is to make some inroads into exactly what we discussed at the very beginning of this lecture, which is how we tackle climate change and how we contribute to the restoration of biodiversity in our built environment. Now, we've had a number of years now of graduates. It's funny because when we started giving these talks, obviously, we were very much an emerging field, and it's been wonderful to see just how some of our former graduates have taken off and really gone on to very interesting and rewarding careers. So not only have we had people participate in exhibitions and conferences, but people have secured jobs with companies working in the field of biodesign, if you like, companies such as Faber Futures. We have others who go on to architectural practices, but equally we have MSc students who go on to either further research, to doctoral studies, or they take a slightly different route and they begin to get into fields that they couldn't have explored before. So for instance, Julian, one of our former students, now works in communication and graphics with the Microbiology Society. So really making use of his design skills to convey some of the more technical aspects in original manners. And on the right-hand side, you can see some of the external engagements just from the last year of the program. We're an incredibly busy and active cohort who've been selected for conferences, nominated for awards like Natalia and a 25 under 25, but also, again, getting out, getting to meetings, exhibiting it or submitting things for biennales, and again, promoting this idea that biology and design and architecture can be very much integrated.
We do give a lot of importance about what students do after the program. So of course, it's great when they're here and all the exchanges of thought and all the designs and making, but ultimately it's about what students take from here somewhere else. Therefore, we do strongly recommend and support students to reach out. And that's what you just see in 1 year, how many students have simply taken the initiative proactively to participate in conferences. Now, sometimes it happens that somebody in BioID just goes on to the stage and, and it becomes a big moment. And this was Shnil Malik at the TEDx Talks Gateway in Mumbai, speaking in front of an audience of nearly 5,000 people. and being seen by hundreds of thousands of people simultaneously. And that's quite special because it just shows the dissemination power and the sheer interest that currently is out there, and the need for new solutions and ideas. So we do simultaneously, while students proactively are participating in conferences and taking this work out of BioID, we're also doing our own research and try to involve as many students as we can, because that's where we have opportunities to scale up. This was an exhibition, the Calcareous Arabesque, where we worked with Tate and Lyle, the sugar company. We developed a new material composite, which was then calibrated for robotic extrusion. And we created this very large 2x4 meter wall for the Waste Age exhibition at the Design Museum in London. Yao Meng was really the guiding force behind the project. She spent, many hours, days, and some months in a row on calibrating and working on these robotic extrusions.
And this is a more recent project which has just been installed out in East London at a site at a place called Barking Riverside. So one thing that we're interested in is the idea of temporary landscapes, how we can also create landscapes, but equally work with materials that are from a place, that have a sense of place. This is a project on impact printing of soil, looking at soil-based construction, not for inhabitation, or at least not of human habitation, but to activate a site which is not going to be built on for several years, but also to enhance the remediation of the soil and to hopefully improve soil health as well. So the idea is to create active landscapes that engage with the environment, but also act as, if you like, refugia for some of the species and native plants. So this was created, again, with this interface between material development, but also robotics and computation for the design.
Also, at the same time, very recently, we have built this little construction at St. Andrew's Botanic Garden. And what is very special about this is that these novel engineered living materials that Prantar has developed, and also the composites with melanin that Ella Hetherington has worked on, they are both now integrated in the design of this little construction, the bioscope. So really scaling up, integrating novelty in a construction that in itself has a certain aesthetic and technical quality in the landscape is really important to us. And that's how we see BioID, as a sort of collaborative environment where we all work with each other, because the different opportunities that come up really are important for us to embrace, because what we need is case studies. We need role model buildings and infrastructures and prototypes that really can show us the next path for the future. These are just two last slides. This is the Indus project that Shneel Malik was leading, and Brenda was highly involved with bioremediation. In fact, this is an interesting case because the bioremediation research was done by Brenda already many years before in biochemical engineering. There were certain research papers, but there was no design. There was no way of integrating this with ceramic tiles. And this is where it became really powerful, because there was an interface where this started engaging the public. And we at the same time have with these bioreceptive walls build several prototypes. This is at the St. Anne's Catholic Primary School in South London, where we were transplanting moss here onto the wall, and we are now monitoring how these walls are weathering and aging, which is really important. Time really matters. And hopefully this animation is running. This was done by a team of students in the first years of BioID, but we still think it really speaks for the entire cohort and the visionary ambition in the program. Thank you. That was a quick, short introduction. Maybe it's worth saying just at the end that we are interested in a really diverse group of students. We want students from many, many different disciplines. We have over 25 nationalities in our program, coming from different parts of the world, from pretty much every corner of the world. And students, yes, there are quite a few architects, but urban planners, product designers, was a pharmacologist, computer scientist, engineers, bioscientists. And it's this mix that is really enriching. This is the one that really makes students feel happy to be here because they're part of a greater scene where they learn so much from each other. And the other thing to say is that in BioID, we are very, very keen that the design is really at the highest level, and it's really competing with the best schools of design internationally. But the same happens with the science. The science that is done here is at the top level. That's why biochemical engineering and the Bartlett have joined forces to work at its highest in each of the departments together. And that's the challenge. That's the most beautiful challenge we face here. We don't try to oblige the other to do what one is doing. They both have relevance and they both have enormous strength. So we hope you can join us. And maybe Nina, you want to say something at the end?
Yeah, I would just add that now it's time for questions, obviously, but you might have some other questions.
I think you're on mute.
Can you hear me?
No, hang on. Sorry, no. We are actually. Sorry.
Sorry. No, just wanted to say that if you have any questions later on, feel free to directly write to me and I will be glad to reply. And any questions about the program and also some others if it's just related to our admissions process. So I'm here too, and I already recognize some of the names that I have been in touch with already. So yeah, feel free to contact me directly at any time.
Stop sharing.
Stop sharing. That's the one. Yeah.
Okay. All right. And it's super exciting to see people join us, presumably from all over the planet. One question we often get asked is sort of what are we looking for? What is it that what we think is good in an application? I have to say, although we do interviews, what we're really trying to get to understand is what happens beyond your CV. Who are you? What are you interested in? What are the kind of curious things that you might be passionate about? What kind of skills do you have that are maybe nobody else possesses? Because what we're trying to see is how do we build the most diverse and interesting cohort of proactive individuals? And proactivity can take the form of many things that may lie outside of your academic career. So that might be hobbies that you do, travel that you engage in, volunteering work, whatever it might be. It's great to see people who are interested in growing plants, cooking food, whatever it is, outside of the sort of normal mainstream activities. And that helps us get an indication of the kind of person you are and whether or not you'll also enjoy an exploratory program like BioID, where really to get the most out of it, we do expect for you to be proactive, to take the opportunities that the city, the institution, but also, you know, the environment at Here East permits you to get involved with.
There is a question here about how many students there are on the course. We have between 40 and 45 students in the 2 years together. So it varies slightly, but I think this year we have 44 students. Last year we had 42. And that means that we have roughly 20 to 22 students in year 1 and the same amount in year 2. The other question here is about what makes an application stand out. It's very difficult to say, to be honest, because we are interested, as we said, in diversity. And we want, as Brenda said, students who are very proactive. who take the initiative of doing things. But we also want, of course, good designers. We want good scientists. We want people who were great in what they were doing and can contribute to the scene, who come with ideas. Now, the way that you want to put this together in a portfolio or in a written form, that's completely up to you. You will see once you apply that there's a little exercise we ask students to do. This exercise might change this year again slightly. But the point is here that we want you to be very honest about how and what you want to do when you come here and how you want to contribute. And that's important to us, because ultimately we want this diverse group of people to work together. And we do a selection that is to do with how we imagine the group can get along, can learn from each other, how they can really benefit from the presence of the others. I don't know if there's any other question that we have here.
Have you noticed that alongside them, students are keenly excited to each other? Yes, I think it's a question from Gigi. This is actually one of the most important things. I think there's two things about this. One is feeling empowered and confident enough to share your knowledge, whatever that might be, and it could be from your academic background or elsewhere. But it's also a generosity. And that's, I think, if we're going to enter, I think what we call the age of the Biocene, what happens next after the Anthropocene, we need to embrace that kind of spirit of kindness, of generosity. It's not just about individual fame or me and mine and getting my stuff. It's spending time with other people to bring them into your world, share some of your experiences, but also maybe your technical know-how. And that time in the studio is vital for that, to create a studio culture that peer-to-peer, but also with the year above, that you're sharing these kind of interesting non-formal things that actually stay with people way beyond the program and start to build a network. Scholarships, yes, we always get asked about scholarships. The Bartlett Promise Scholarship, I believe, is something that's open to those who meet the eligibility criteria. Last year, we were able to offer one full scholarship through a sponsorship for an MSc student via a collaboration with PGIM Real Estate. We have to confirm whether or not we're going to be offering that again this year, but if we are, we will be able to publicize that and make you aware of the application criteria. But yeah, as I said, probably the best source for information on scholarships is either, we have many people who come actually from their home countries with scholarships, and we're always happy to support applications of people in that manner. But yeah, at the moment, the best source of scholarships is probably via the Central Bartlett website.
I'm reading here the question about how collaboration with multiple research groups happens. So first of all, the internal collaboration is that students work individually in certain parts of the year and in certain modules, and they work collaboratively in others. And that means that their personal interests that students develop and certain types of skill sets and a certain expertise that they are really keen on, and we give students that space. At the same time, there is no interdisciplinarity without collaboration. And that means that we make sure that the MSc students and the MR students really work together, intermingle, so that there is this cross-pollination. And we do suggest students with different interests to come together. That's something that we spend quite a lot of time doing, just make sure that the different teams are evenly distributed, so that there is different knowledge and expertise and different methodologies coming together in how students work. It's not easy in a sense, because in some cases we leave it up to the students to decide. More initially, usually we decide, because they don't know each other yet that well. Now, these collaborations can be explosively interesting, as you can imagine, when a bioscientist, an architect, and a product designer, let's say, are working together. We do have then, through the thesis report, this link with external people who come to supervise certain projects. That's where potential collaborations with other groups might happen. And that's where this external input and feedback becomes really important. We as a program, I think we offer a lot, but we can't offer everything. It's just obvious. So a lot of it is covered by those external tutors who we bring in for the thesis report for these several months where students are working on a particular type of project. Is there any other question here?
Yeah, there's a question about the types of organism. We work with, well, so far we've worked with bacteria of all kinds, algae, plant tissue culture, fungi. I mean, apart from the archaea, I think there's really no kingdom left untouched. It's the lab setup that we have here is really able to grow most things, photosynthetic or non-photosynthetic. And then we have access to biochemical engineering if there are projects that really go beyond, that require a sort of level of specialism or a level of equipment that that merits perhaps some extra or some more specialized facilities. But we have people who've worked on enzymes, we have people who worked on maybe perhaps looking more at circular economy and sort of almost biomaterials rather than the living. So there's a whole spectrum of things that people engage with, but the most important thing is in term one, you get the opportunity to grow things for yourself and start to experiment.
There was, as part of Ben's question, any advice for the high schoolers? Maybe that's for others. Well, to be honest, to be a great observer and have curiosity is sort of maybe are the two most important characteristics. If you really want to learn more because you're curious, you start observing what's happening around you. Of course, you will read into what others have written and done, and you will do your own experiments. But we have an outdoor laboratory, which is fabulous, which is the world. The world is a phenomenal laboratory where things are happening. And to be vigilant, observant, and then deduct certain rational processes to understand what's actually happening is in itself incredibly important. And the other thing we often say to applicants is when they ask us how to prepare, they say, have you grown anything? Just to grow something and maintain something growing is in itself quite a big challenge. So that will imply how much water you put on it, you know, what the environmental conditions are, et cetera, et cetera. So that is not just trivial. It takes a level of expertise to really do it in a more diligent manner.
It's true, and it's funny, people often feel like, oh, but I can't get started. Well, guess what? Many people who live in urban environments don't have access to green space. If you don't see that as an interesting design brief from the very beginning, yeah, this is really a program that tries to tackle some of that and look for ways to innovate. So don't see it as a limitation, see it as perhaps an opportunity for you to do something creative in your own home. I wanted to quickly address a question about comparing ourselves to other institutions. Look, it's these other institutions are phenomenal places where there's really interesting programs and really interesting people. If anything, I would say we are spoiled in London because we have an environment where there's lots of people, lots of intensity, and that's good, actually. You want the people around you to be great because it's stimulus for you. It's great to see their shows, great to see their exhibitions. Now, the one thing I would say is UCL is a very different type of organisation to Central St Martin's in that we have faculties of engineering science, we have institutes of structural and molecular biology. So we have the kind of deep core of the science landscape as well. So for the MSc students, the program is run, or degree is run awarded conjointly with the Department of Biochemical Engineering. But day to day, what you're able to feed into is that kind of deep-seated knowledge where we have interchange or exchange with postgraduate teaching assistants, a research landscape, and access to just some of the most phenomenal academics who perhaps join as thesis supervisors or come and give seminars. So it's not just about labs and facilities, because actually I think to do the most interesting science isn't really dependent on the most interesting gear. I actually, in all my life, in all the institutions I've worked in, I can say quite confidently, it's not how new your equipment is, it's how good your ideas are. As Marcus was saying, can you engage with the big laboratory that is planet Earth? But it's the environment that we place ourselves in and situate intellectually and academically at UCL amongst some of really just the finest people who are doing really cutting edge stuff in earth sciences, in molecular biology, synthetic biology, and engineering.
The other thing to say is that one of our big strengths is and focus is we are a design program with the MSc and the MArch route. We do science, we do material experiments, but we design and we do scale up. And one of the great advantages of being in Here East is that we have high ceilings and equipment and forklifts and etc., which allow us to operate on a level that not even our colleagues in the other Bartlett building can operate in. And this is to do with really being able to scale up and to think about the build environment by and large, including, as I said before, what a piece of furniture might be in it. So we don't force anybody to design buildings, but we are in the top #1 school of architecture, and therefore the design ethos that we have at the Bartlett with all its diversity obviously plays a major role in how students develop their design work here. Is there any other question? There's an BArch student, so Bachelors of Architecture. Ariane, if there are any tools, look, you can learn a lot of software and do lots of environmental studies. In a way, the interdisciplinarity doesn't have a recipe. The interdisciplinarity needs a method that you gradually develop by yourself with colleagues, and it needs a certain context. That's why BioID was created by both of us, because we were doing things individually and we knew a few colleagues who were doing these things, but there was no context for these things to be done. We were doing them pretty much in isolation. Now, of course, there's lots of environmental software, analysis software that you can use. That's interesting. There's also, there's certain programs, computational programs that allow to simulate growth patterns. That's also interesting. As I said before, growing plants is in itself actually much more challenging than what it looks like. And to start combining these becomes very, very, very difficult and fascinating. And anybody who makes the attempt will discover the limitations of extrapolating concepts and techniques from field to field. The workflow is still a difficult one, but that's what we're working on. That's where the future is lying, is to create a workflow where we can go from a laboratory into pure computational simulations and doing material studies, and then scaling up and scaling down, and have that in a continuous loop. We do it, but we have still to extrapolate lots of ideas and concepts. But that's maybe also where the opportunities lie to critically think what we are doing and analyze the results and then take them further.
Yeah, and I would say, further to sort of growing a plant, strengthen your skills in biology and observation, get out and volunteer, do some species recording, volunteer at a garden, look around you for opportunities to engage with citizen science wherever you are in whatever country you're in. I would say probably my personal scientific journey started through citizen science as a way in, and it invites you to observe, to record, and also to discover things about the world around you. I think nowadays there is a huge amount of open source information, places like Materium and, you know, platforms that, you know, give you recipes for biomaterials. I wouldn't be fixated on trying to replicate everything that you see out there. I'd be much more interested when we meet you at interview to see what you're doing that's very particular to you, and your interests, and whatever those might be, however esoteric you might believe they are, I can assure you they're much, much more fascinating to us than merely seeing you recreate or be inhibited by the kind of work that's already made. We're interested in the ideas that haven't been made yet and how we can help you to realize those on a program like BioID.
By the way, there is something very important we were not mentioning
What was that?
to everybody, which is to visit some really good buildings.
Oh, yeah.
There are
Okay.
far too few students nowadays, even in an architecture program, who have visited really lots of amazing buildings. This experience is really fundamental for the understanding of what makes certain structures, spaces, and environments be really extraordinary and game changers in their time. So this experience, that's why we do the field trips, is important because it generates a discussion. How even something that was very pivotal just 50, 60, 70 years ago might not have great relevance right now, or something that was almost overlooked 100 years ago might be incredibly relevant in this point in time. And these historic references do matter to us. They make us think and contextualize and make sense in a more multi-layered manner how good projects, meaningful projects really might create change.
Yeah, I would say, and if I was an MSc student, I wish I was a future MSc student, it'd be great fun. But if I was a future MSc student, I would also maybe get out and practice my drawing skills and learn to it's not learn to draw, it's learn not to be shy about expressing yourself through image making. And a lot of MSc students spend a very long time sort of saying, well, I drew something, but I don't want to show it to you. And I think if you have the time now to get comfortable with drawing, making, image making in whatever way that you find interesting or relevant to you, it's to cultivate that personal practice. And don't wait till until you're told to do it, but to realize, and even if you don't apply to BioID, which is absolutely fine too, well, we'll miss you, but if you don't apply to BioID, but to realize you have full permission to do these things in your spare time. You don't have to show anyone. You don't have to, you know, worry about whether or not you think it's comparable to what someone with 6 years of architectural training can do. You just have to get out there and do it and have that freedom to experiment and realize that, yeah, the world is your laboratory, but it's also, and there's many things that you can do to try and interpret it for yourself. And drawing is, I think, a really interesting one.
I think we are just over an hour. Thank you for having been with us for this short introduction. If you have any other more relevant question, please send to us. Nina will be mainly in touch with you for the admissions process or any other question. And we look forward to seeing you hopefully here at some point. It would be amazing. And to start this journey together. Yeah. So enjoy, if you're in the final year of your studies, make sure you enjoy that year. And you're probably looking ahead, but also make sure that you're fully present in the research projects and whatever else that you're doing. And yeah, best of luck with everything. And I hope we do meet you at some stage here in London. Thank you. All right. Thanks for joining us. Bye-bye.